Hi everybody, my name's Steve Wakelin, Research Leader at Scion Research, and today I've got the privilege of talking to you all about the pine microbiome projects that we've got happening here in Scion. So let's just go into the share screen here. Stop the video. So hopefully that's coming through quite well. So the title of my talk is The Role of the Tree Microbiome, and that will become clear as we go through the presentation. So this is certainly on behalf of myself that's presenting, but also the pine microbiome research team in Scion. So a really good question that we should start off with is, what is a microbiome? So microbiomes refer to the entire community of microorganisms that live in a defined environment. So it's the assemblage of all the species that are there in certain habitats. What's really interesting is that each habitat, whether it's a soil or a plant or a person, has its own unique fingerprint of the microbes that are present. So microbiomes can be incredibly diverse and complex in species and functions. And this picture is Winogradski's column. It's a very good example of this. Lots of gradients of sulfur, oxygen, and energy means that there's lots of different habitats and spaces for the microbes to live in. So microbiomes can also be very simple. And a good example of this is the microbiome of gold grains. So yes, microbes live on gold, and in this case, there's only one or two species that are very common in these habitats. It's very simple. So microbiomes aren't just out in the environment in the world, they're also in and on us. And so people will, often talk about the Human Microbiome Project, which was started in the US a few years back. And that was really to try and characterize the assemblage of species of microbes that make up a human being and affect our health. So there's something like 100 trillion different microbes that live on or in each person. What's really cool is that for every one human gene, there's something like 150 microbial genes, systematically expanding the catalogue of genes that make up a person. And each individual person has their own microbiome. And that's as unique as a fingerprint in terms of the species that are there. And another important fact is that what we know now, unraveling the human microbiome, tells us that something like 90% of diseases have some sort of link back to the microbiome. So these aren't just diseases associated with your obesity and weight and all the rest of it. It's neurological disorders right through to Parkinson's disease and so on. They've got a microbiome link, which we never knew about until we started this work. So plants too have involved with unique microbiomes. They're in and on the plant surfaces. That includes the soils and the roots, right through to the leaves and the other plant structures. And they're functionally important. They're not just microbes sitting there doing nothing. They have a role to play in the success and life of the plant. And some good examples are how microbiomes extend the phenotypic plasticity or elasticity of a plant and environment. A good example is drought tolerance. Microbes can confer drought tolerance in some plants. The microbiome can change the growth rate of plants, whether that's positive or negative. Of course, we want it to be positive most of the time. The microbiome has a role in disease, causing disease, and also disease suppression, nutrition, right through to regulating the onset of flowering in certain plant species. What's also sort of coming through the literature at the moment is the role of the microbiome in and on the plant and sort of conferring new metabolites and secondary metabolite pathways, which sort of changes what the plant does. And these are really, really important. So these are like flavor compounds in strawberries, or the unique factors in honey. New Zealand's agricultural sector, the ryegrass, is dependent on a microbiome to protect it from the grazing of invertebrate pests. So they're really, really important. I suppose it's not surprising, therefore, that there's really a lot of international investment in understanding and harnessing the microbiome of plants. So here's one example of, Monsanto and Novozymes getting together to form the BioAg Alliance, which is something like $100 million investment in understanding or hacking the microbiome of agricultural plants. This means that they're really trying to reduce the amount of nutrients that plants need to grow, and right through to protecting the plants from diseases and pests. So what about trees, and what about Pinus radiata? So trees should really be the priority research for plant microbiomes. I mean, trees have long breeding cycles, so it can take quite a long time to introduce new traits just by the standard selection and breeding approaches that we have. Because they're in the environment for a long time, once you plant them, they're sort of staying there. Trees also require a large degree of environmental elasticity to future conditions. The conditions that we plant them in today are probably going to be quite different in 20 to 30 years' time. And this is not just the biophysical conditions of the environment, but things like diseases and pests, and maybe the way that we manage forest as well. So what we're proposing is that, like with the human microbiome and other environments, the microbiome of a tree is really a new genetic library that we can add to the tree to confer new traits. And whether this is around productivity gains, stress tolerance, environmental resilient forests, wood formation and quality, there's lots and lots of opportunities there, and we haven't even really started exploring these. So the tree microbiome research, what we aim to do is look at the plethora of the microbes that are on and in a tree and the environment that they're in, and particularly the interactions and the outcomes of those interactions of the microbiome, the tree and its genome, and the environment, all of them together, and how they sort of function to alter the plant physiology and the growth, and even the behaviour, the coordinated behaviour amongst the trees and the forests, and how they sort of change the whole ecosystem state of all the geochemistry, so the nutrient cycling and the carbon storage. Now this is quite a challenge, and we've done a lot of microbiology research, but microbiome research is a whole nother level. And, if English philosopher Francis Bacon said that if we're going to achieve results never before being accomplished, then we must expect to employ methods never before being attempted. Fortunately for us, we've got the Human Microbiome Project and some other endeavours to fall back on, but certainly it's very ambitious to go in and start exploring the tree microbiome. But we do need to start somewhere, because the opportunity size is very big. And towards this, we've got two highly aligned programs of work that we'll just quickly run through. So the first is the Resilient Forest Partnership Program, and the second is some announcement of new government funding. So it's through the MB Endeavour program, a grant called the Pine Microbiome. So most folk will be aware that we've just completed year one of the Resilient Forest Partnership Program, and the role for us in this program in the first year was building the fundamental framework for pine needle microbiome research. So we really needed to get our stuff together to start exploring the microbiome and what it does to the plant and the environment. And we did start with needles because of that direct link between needles and disease and the microbes. And if you're losing your needles and they're not functioning very well, you're losing your solar panels for the plant to grow. So in the first year of this research, we've been exploring field sterile techniques to collect samples from the tree and the forest, returning those back to the laboratory, trying to understand the variability on the tree and the canopy of what the microbiomes are on a needle. So we're in the canopy to select how old the needles are, what really makes a difference when we go out sampling. So, a fair amount of fundamental framework work had to be done. And then in the laboratory, we need to understand, how to extract the microbiome DNA from the needle. So not just the plant DNA, but all the other DNA that's there, and really scale up the way that we do our sequencing. So traditionally, we might have a sample and we sequence them one by one. What we have been able to do this year is massively parallelize this sequencing. So we're typically sequencing 88 samples in one go at the moment. And then right through to the unique bioinformatic and statistical approaches that we need to start dealing with this data. So really, just getting all our ducks lined up. So as we go further into the program, all the basics are there and we can proceed with confidence. So although year one was really about getting the framework established, some really interesting results are already sort of emerging from this work. An example here is the expected amount of fungi, not even the bacteria, just the fungi, that we can expect on the pine needles of a single plant. And there's more than 1,600 species that we've discovered so far. And that's probably the limit for this plant in this environment. Based on the average genome size of the fungi, there's actually 2.5 times more genetic information and genetic potential and novel information just in that genome compared with that of the tree. So if we remember back to the human microbiome slide where every human gene has 150 microbial genes, well, just on this one tissue component alone, we're starting to see a very similar pattern emerge. What we've also found, which was very nice, was that the fungi that are there aren't just in a single small clade of similar species. There's a massive diversity of species. There's polyphyletic distribution of fungi all across the kingdom there. And that means that the range and the potential of traits and DNA and things that these fungi can do it's absolutely immense. So we've got single cellular fungi from yeasts, which are known biological control agents for the phyllosphere, for the needles, right through to the typical branching pathogens and saprophytes that we might expect to see in a forest ecosystem. And just sort of scanning through this tree of the fungi that are present on the pine needles, the sharp-eyed people in the audience will notice that there's a group of the Dothidium mosaics, and this is where Dothostroma, the fungal pathogen, sits within amongst this group. So the needles that we sampled were ostensibly very healthy. That was one of our requirements for sampling. And yet the pathogen, or a close neighbour, is there. So it's sort of silently sitting there and perhaps waiting for the microbiome to change or the conditions to change to express a disease outcome. So already, we're starting to, come up with some new understandings of where some pathogens might be sitting latently in the forest, ready for diseases to emerge. Year two of the Resilient Forest Partnership Program is really about building on the gains of year one. So we've built those foundations, we know what to do and how to do it now. And year two is really extending what we do on the needles and our understanding into the different plant tissues. So looking inside the tree itself, the microbiome in the tree, in the wood, the reproductive tissues, the pollen, the catkins, right through to the bark and cambium. So really the microbiome that's in contact with all these other cells and signaling, that's in a really profound position to change the wood formation, wood quality, growth aspects, hormoning, signaling with the plant and the environment. And this is downstream in 5 or so years' time, really what we want to end up with, where we've built up an understanding of the tree and we understand its genetic potential. We understand what the microbiome is on that tree, the assemblage across the roots and the soil and the trunk and the needles all acting together, and how these are expressed collectively against the environmental conditions. So temperature, silvicultural management, herbicide and pest disease, all that sort of thing, how they are expressed against that entire tree and its microbiome to affect things like carbon storage, how to climate-proof forests, how to control pests and diseases or confer tolerance, how to alter wood quality or store carbon in soils. So really for us, it's taking the next step in our understanding of how forests operate and function. So the other program that I'd just like to announce is some new funding that's come through from the New Zealand government into the forestry sector. So it's an announcement of investment of $13.5 million by MB, and this has been supported by the Forest Growers Levy Trust, and we're very thankful for that. So it's a 5-year research program. It's been led by the team here at Scion, but it's certainly multi-institute and international in flavour. And what we're going to do is use Pinus radiata to determine how evolutionary history, how pine co-evolved with microbes to be successful in the past, how genetics, how the ecological context all influence the outcomes of the root microbiome interactions, and extend the tree phenotype. And the target in the project is around climate proofing forestry, but really that's just the one of the many things that could potentially come out of here. So a key outcome for us is the development and establishment of Pinus radiata as a model tree root soil microbiome platform globally. And the reason that we want to do this is that our resources and the amount of time we have available to make these gains in New Zealand is quite limited, and we want to be able to harness off what people can do globally. So if we establish pine as the model, like in a crop species that other research labs take up all around the world and publish on, well, they're essentially doing a lot of the work and investment for us, and those benefits, those science outcomes and how we interpret them for New Zealand forestry will flow through, so we can get, it's a case of working smarter, not working harder. Another key outcome will be the demonstration that targeted manipulation of the microbe on a certain tree or a certain cultivar confers reproducible changes in the physiology of that tree. So really demonstrating, without a doubt, that manipulation of the microbiome can change the tree in beneficial ways. This program that the government's investing in with the Levy Trust is explicitly focusing on soils and the soil root microbiome. And this is a really good outcome, because what that means is that it's directly complementing the work that's happening in the Resilient Forest Partnership Program, which is really above ground tissues. So that these two programs really line up really nicely. And to us, this is really an investment in the next generation of microbe silvicultural tools. So in some ways, we, like the Human Microbiome Project, we might not know what's going to come out at the end of this, but there'll certainly be some incredible surprises and opportunities. And in the future, we hope to be able to, use the microbiome as another one of the tools that we've got in our silvicultural toolbox, and the plant genetics and genomes and things as well, to manage sustainable forests for New Zealand's future. So that's the end of the talk. And I've just noticed that there's a mistake on there. There should only be one @ in that email address, but I'll leave you there with that. And thank you all for your time and attention. And I just sort of recognise that timberlands have been really great to us, enabling access into their sites and getting their plants to push this research along. So thank you very much.